So, the immune system, how does our body respond to disease specifically? Well, the immune system is a large network of organs, white blood cells, proteins, and chemicals that protect the body from germs and other invaders or viruses. The primary functions of the immune system is to fight off bacteria, viruses, parasites, and fungi that enter the body, identify and respond to antigens, which are substances on the surfaces of cells, viruses, fungi, or bacteria, to fight disease-causing changes in the body, such as cancer cells, to limit the amount of harm that invaders can cause if they're inside the body, and to heal damage to the body, for example, cuts, bruises, or bone fractures, and prevent infection of these injuries. So, what type of feedback system is the immune system? Well, it can be both in different scenarios. So, it can be a negative feedback system because it's making sure that the immune system doesn't go overboard and harming normal cells by producing too many immune cells. For example, infection could bring a lot of inflammation, causing immune cells to respond continuously to the infection. However, certain other cells would make sure that inflammatory cells don't go too far and harm normal cells. It could also be a positive feedback system because if there is an infection, the immune cells in that area continuously bring in new immune cells to deal with the infection, which pushes it out of equilibrium. So, the immune system is made up of two different parts, the innate immune system and the adaptive immune system. So, first, the innate immune system provides a general defense against harmful germs and substances, so it's also called the nonspecific immune system. It mostly fights using immune cells such as natural killer cells and phagocytes, or eating cells, and the main job of the innate immune system is to fight harmful substances and germs that enter the body, for instance, through the skin or digestive system. This nonspecific mechanism is fast, while adaptive response takes longer. And the innate immune system can be split into a first line and second line of defense. So, how does the innate immune response work? Well, at first, bacteria can go unnoticed, but when a certain bacteria population is reached, they damage the body by changing the environment around them. First, macrophages, or guard cells, intervene. These are huge cells that guard every border region of the body, and they can swallow up to 100 bacteria, and each of these are broken down by enzymes in the cell. They cause inflammation by ordering water into the site to make fighting off bacteria easier, and this causes mild swelling. Next, these macrophages release messenger proteins to communicate the location and urgency of the bacteria or infection, and these messenger proteins call neutrophils to leave their normal position in your blood vessels and move to the infected site, where they will kill healthy cells in the process of trapping and killing bacteria cells. And like I said before, they have a short lifespan and normally will kill themselves after around 5 days, so they don't do too much damage to your own body cells. And complement proteins are flushed in with fluid or water, causing the inflammation as well into the infected site. And next, the dendritic cell becomes active after being notified by either PAMPs, which are pathogen-associated molecular patterns, or DAMPs, damage-associated molecular patterns, inflammatory mediators, cytokines, or cellular stress molecules. And the dendritic cell starts collecting samples of the bacteria and covers themselves in it, and these decide whether to send antivirus forces or antibacteria forces. This dendritic cell will travel to the closest lymph node where billions of helper B and T cells are located and waiting to be activated. And this dendritic cell will look for a specific helper T cell to bind to the ripped off piece of bacteria on its outer membrane. And moving into the adaptive immune response, the adaptive immune system makes antibodies and uses them to fight specific germs the body has previously encountered. It is constantly learning and adapting so the body can fight bacteria and viruses that change over time. And some things about this, it responds in a highly specific way to the pathogen that caused it, meaning that the body will later remember the pathogen, which allows it to respond more quickly and effectively the next time it encounters that pathogen, which is why you can only contract some illnesses once, because your adaptive immune system does such a good job that you don't even feel the symptoms of said virus or bacteria. It is much slower than the innate immune response, but is much more accurate. And you're not born with your adaptive immune system. It instead grows as you encounter more and more microbes that your body can learn to defend against. T cells can rearrange and reassemble pieces of DNA to create billions of different receptors that DNA recognize and help attack every different infection or bacteria that you face. So, to go into how this actually works, after the dendritic cell covers itself in the piece of bacteria and moves its way into the lymph vessel and finds the specific T cell with the matching receptor, it will activate it and link up with it, and the activated helper T cell will duplicate rapidly after the chemical reaction, and some will stay in the lymph nodes so that you are immune to the bacteria next time it presents itself, which are memory T cells. Some travel to the site to help fight the infection, which are killer T cells. Some travel to the center of the lymph node to activate the virgin B cell with the same setup as it, which is helper T cells, and then the suppressor T cells will also stay around in the lymph nodes to turn off the immune system after it is done and the immune cells are stop getting stimulated. Then once the T cell finds the virgin B cell, it activates it, and the B cell will duplicate rapidly, which produces small protein antibodies that will bind to the surface of these specific intruders. And helper T cells communicate to plasma cells what type of antibody is needed in the specific infection. Billions of antibodies are sent to the site of infection to either kill bacteria or connect to the infected cells to allow other immune cells to attack and kill them more easily. Without constant stimulation by helper T cells, the macrophages, killer T cells, neutrophils, and other B cells kill themselves to stop doing damage to your own human body cells, and only memory T and B cells stay alive in your immune system if your body gets attacked by the same infection again. So that was a pretty generalized explanation. Here is a diagram of just all of the immune cells and how they're all connected. You can see in the top right hand corner here how they're all connected by the different colors. You can take a look at that later if need be. And to get a little bit more specific now, looking at the immune system's monitors, coordination centers, and regulators. These are things that I didn't understand fully when I was doing my original research, but after preparing for this presentation, I went a bit more in depth with my research. So the monitors of the immune system are its surveillance mechanisms, constantly patrolling the body to detect pathogens, abnormal cells, or tissue damage. These include specialized cells, receptors, and signaling molecules that act as the system's first responders. Phagocytic cells, such as macrophages and neutrophils, play a crucial role by engulfing and destroying invading microbes or debris. These cells use pattern recognition receptors, or PRRs, like toll-like receptors, TLRs, to identify pathogen-associated molecular patterns, which are called PAMPs, which I mentioned earlier, and damage-associated molecular patterns, DAMPs. Dendritic cells also serve as key monitors as they patrol tissues, capture antigens from pathogens, and migrate to the lymph nodes to present these antigens to T cells, initiating the adaptive response. The immune system relies now on the coordinating centers to organize and direct its responses, ensuring efficient defense against pathogens and maintaining balance. Key coordinating centers include the lymphatic system, which connects immune cells and facilitates communication. Within this system, lymph nodes act as checkpoints where antigen-presenting cells, or APCs, like dendritic cells meet T and B cells to activate and tailor adaptive immune responses. Similarly, the spleen filters the blood, removing pathogens and damaged cells while organizing systemic immune responses through its white pulp, which houses lymphocytes. At the mucosal surfaces, or the MALT, mucosa-associated lymphoid tissue, including structures like Peyer's patches in the gut and tonsils in the throat, serves as a local defense hub, coordinating immune responses to inhaled or ingested pathogens. And beyond physical structures, soluble molecules like cytokines and chemokines play a crucial role in coordination by acting as chemical messengers. And cytokines amplify and guide immune cell activities, while chemokines direct immune cells to infection sites. The thymus also acts as a specialized coordinating center during T cell development, ensuring that T cells recognize foreign antigens but not body's own tissues. Additionally, the bone marrow produces all immune cells, providing the raw materials for both innate and adaptive immunity. Together, these coordinating centers ensure that the immune cells interact effectively, respond appropriately to threats, and maintain overall immune balance, or homeostasis. And going into the regulators, regulatory cells are essential components of the immune system that maintain its balance by preventing excessive activation and promoting tolerance. Among them, regulatory T cells, or TREGs, play a pivotal role. TREGs suppress immune responses by secreting anti-inflammatory cytokines and by directing inhibiting effector T cells, thus ensuring self-tolerance and preventing autoimmune diseases. Similarly, myeloid-derived suppressor cells, or MDSCs, a diverse group of cells, regulate immune activity by suppressing T cell responses, especially in conditions like chronic inflammation, cancer, and infections. Regulatory B cells, or PREGs, also contribute to immune regulation by producing certain cells to control excessive immune responses and reduce autonomy. And lastly, dendritic cells can adopt tolerogenic properties under specific conditions, promoting the differentiation of TREGs and limiting the activation of effector T cells. Collectively, these regulatory cells work in concert to prevent and maintain immune homeostasis. Collectively, these regulatory cells work in concert to prevent immunopathology and maintain immune homeostasis. Alopecia areata is an autoimmune disease where the T lymphocytes around the hair follicle attack the hair follicle, causing the patch of hair to be infected and fall out. This affects the hair growth cycle of anagen, catagen, telogen, and exogen phases, and alopecia areata occurs when hair follicles in the anagen phase prematurely move to the catagen, telogen, and exogen phase. So, to connect this back to the biochemistry unit, first I want to look at proteins and enzymes. Obviously, proteins and enzymes are fundamental to cellular function and immune responses. Enzymes such as kinases and proteases are involved in signaling pathways that regulate immune activity. In alopecia areata, proteins like cytokines are overproduced, signaling an immune attack on the hair follicles. Enzymatic activity linked to inflammation and antigen presentation, such as MHC, major histocompatibility complex proteins, is critical in the disease's pathology. And surrounding DNA and RNA, DNA holds the genetic blueprint, while RNA helps produce proteins via transcription And translation: genetic predisposition plays a significant role in alopecia areata, and research into microRNAs, which are small non-coding RNAs, shows they regulate gene expression involved in hair follicle health and immune responses. MicroRNAs can silence or enhance the expression of genes involved in the immune responses and hair follicle maintenance. For example, dysregulated microRNAs in alopecia areata can affect pathways controlling T cell activation, cytokine production, and apoptosis in hair follicle cells. And going into lipids and the cell membrane, lipids form the structural basis of cell membranes, influencing cellular communication and signal transduction. Disruption of lipid signaling pathways in immune cells, such as T cells, may contribute to the autoimmune attack in alopecia areata. Additionally, damage to the lipid-rich membranes of hair follicle cells can result from oxidative stress during inflammation. And lastly, looking into enzyme recognition and inhibition, enzyme activity can be regulated by inhibitors or activators, and therapies for alopecia areata often involve enzyme inhibitors such as JAK inhibitors. So going into metabolism, the energy demand in immune activation. The immune system has high energy demands during an immune response, and processes like T cell activation, cytokine production, and antigen presentation require significant ATP, which is generated through cellular respiration. In alopecia areata, overreacted T cells attack hair follicles, and this immune activation relies on energy generated by aerobic glycolysis. And the switch to glycolysis, even in the presence of oxygen, is a hallmark of activated immune cells, contributes to the persistent inflammation seen in alopecia areata. Also, the mitochondria play a central role in metabolism, obviously, energy production, and cellular health, and mitochondrial dysfunction has been implicated in alopecia areata, as it can impair the energy supply to hair follicle cells, disrupt normal cell function, and trigger apoptosis, which is programmed cell death. And this ties directly to the metabolic concepts of ATP production and energy transfer. And metabolic pathways such as protein synthesis, lipid metabolism, and glucose utilization are essential for cellular growth and repair. And hair follicle cells are highly metabolically active, requiring constant energy and nutrient supply. Dysregulated glucose or lipid metabolism, often influenced by inflammatory cytokines, can impair hair follicle growth and regeneration. Lastly, looking at amino acids and immune function, the amino acids are not only building blocks of proteins, but also precursors for important metabolic intermediates and signaling molecules. Immune activation in alopecia areata can alter amino acid metabolism. Specific enzymes induced by inflammatory cytokines break down tryptophan into carnitine. This pathway affects T cell activity and has been implicated in the immune dysregulation seen in alopecia areata. And lastly, looking at molecular genetics, first of all, gene variants regarding autoimmunity. Gene mutations or polymorphisms can alter protein function, leading to disease. Inheritance patterns show how genetic predispositions are passed on. Specific gene variants are strongly associated with alopecia areata. Some of these genes encode proteins that present antigens to T cells. Mutations or polymorphisms in these genes can lead to improper antigen presentation, triggering an autoimmune attack on the hair follicles. Gene expression involves transcription, DNA to RNA, and translation RNA to proteins, and regulatory elements like promoters, enhancers, and transcription factors control this process. Dysregulated expression of genes involved in immune responses, such as those encoding cytokines, contribute to alopecia areata. These cytokines amplify the autoimmune attack. Additionally, altered regulation of immune checkpoint genes affects T cell activation and tolerance, driving autoimmunity. MicroRNAs or miRNAs, like I mentioned before, are small non-coding RNA molecules that regulate gene expression by binding to mRNA and preventing translation. Research shows that specific microRNAs are dysregulated in alopecia areata. For instance, some microRNAs may suppress genes that maintain immune tolerance, while others may enhance inflammatory pathways. This post-transcriptional regulation can exacerbate hair follicle destruction. And lastly, genetic inheritance. Genetic patterns explain how genetic traits, including predispositions to certain diseases, are passed from parents to offspring. Alopecia areata often shows a polygenic inheritance pattern where multiple genes contribute to susceptibility. Studies have identified that genetic loci, which is a specific location on a chromosome where a gene or genetic marker is found, linked to alopecia areata, such as those involving immune system regulation and hair follicle development. And these were all my resources. Like I said before, I wanted to do a bit more research than I did for my original findings because I felt like I did not go in depth and in detail enough. And I apologize again for the mispronunciation of any of the words that I used. Thank you for listening to my presentation.